It's a great honor to speak to you all today about a subject near and dear to my heart, which is oxygen. And we require many things in this life, but nothing more than oxygen. And if you dispute that fact, please hold your breath and I'll get back to you later. The discovery of a system that ensures that every one of the 50 trillion cells in your body receives adequate oxygen at every moment of every day for your entire life. And this is basically a very simple matter of supply and demand. The cells use oxygen to generate energy in the mitochondria, and then we have respiratory and circulatory systems to deliver that oxygen. And the hypoxia-inducible factors that we discovered serve to regulate oxygen supply and demand. If oxygen is not available, the cells use metabolic processes that don't require oxygen, and oxygen delivery is increased through changes in the circulatory and respiratory systems. Well, this was the scene in Stockholm in 2009. You can see the royal family on the right, the laureates on the left, and Nobel in the middle there. This was the King of Sweden giving me the Nobel Award, and this is the award. Thank you very much. On the front, you can see Albert Nobel, his birth date, and the year that he died, which was 1896, and he died on December 10th. And so the Nobel Prizes are awarded on December 10th every year, which is a fairly dark day in Stockholm. But it was also very nice in the winter. And then on the back, you can see my name and the date of the award, 2019. And then in Latin across the top is a quotation from Virgil, which translated into English is roughly, "Let us improve life through science and art." And truly, we've tried to improve life through science. And this started 33 years earlier when we asked a question. We knew that the kidneys produced a hormone called erythropoietin, or EPO. And EPO is secreted into the bloodstream and goes to the bone marrow and stimulates red blood cell production. And of course, it's red blood cells that carry oxygen to all the cells in the body. And the body doesn't count red blood cells. Only hematologists do that. What the body does is sense oxygen delivery. And if oxygen delivery is not adequate systemically, it increases the production of EPO, and that leads to an increase in red blood cell mass and oxygen delivery. So it's a beautiful homeostatic system to maintain systemic oxygen delivery. And when we set out, we wanted to try to understand how do cells in the kidney sense oxygen availability and respond to decreased oxygen or hypoxia by increasing EPO production. And we first identified a sequence that's just downstream of the EPO gene in the human genome that's now known as the hypoxia response element. And this short 33 base pair sequence has the property that it can be taken out of the EPO gene, inserted into a heterologous gene, and the expression of that gene will now be induced by hypoxia. And we use that DNA sequence to identify a protein that was present in hypoxic cells, but not in cells that had adequate oxygen, and which bound to this DNA sequence. And we called this protein hypoxia-inducible factor 1, or HIF1. So a factor is a protein. Inducible means it's made in response to a stimulus, and the stimulus is hypoxia, or low oxygen levels. So this protein HIF1 was the first member of a family of proteins identified that are critical for oxygen homeostasis. And when we looked at the levels of the two subunits of this protein, which we called HIF1 alpha and HIF1 beta because we're scientists, so we like to use Greek letters. We found that the HIF1 alpha subunit was present at very high levels in the nucleus of hypoxic cells, and then decayed exponentially as oxygen concentrations were elevated. And it turned out that the levels of the HIF1 alpha protein were regulated by a very elegant mechanism, which is that when oxygen is available, an oxygen atom is inserted into the protein, and this is a signal for its degradation. So when oxygen is available, this hydroxylation event occurs. It occurs on one of two different proline residues within the protein, and this allows the binding of a protein called the von Hippel-Lindau protein, or VHL. And when VHL binds to the hydroxylated HIF1 alpha, this is a signal for its destruction in a area of the cell called the proteasome. So your body is continuously making HIF1 alpha, and if oxygen is available, it's degraded, and if it's not available, the protein rapidly accumulates within hypoxic cells. So this ability to regulate the levels of the protein is critical for understanding, for example, why erythropoietin is made at increasingly large amounts under hypoxic conditions. So hypoxia, as I mentioned, is an imbalance between oxygen supply and demand. It can be due either to the fact that cells are using more oxygen. So, for example, in a tumor, there are more cells consuming oxygen, leading to a hypoxia within the tumor. Or it could be due to a decrease in oxygen delivery, as occurs with, for example, atherosclerotic coronary artery disease, where the delivery of blood through one of the main coronary arteries is impaired. And in either case, HIF1 is induced, and it turns on the expression within each cell of hundreds of genes. Some of these genes serve to increase oxygen delivery, like EPO. Sometimes this occurs only within certain cell types. So, for example, if there's vascular ischemia, only the tissue that is subject to the ischemia shows an increase in HIF1 levels, and this leads to the production of factors that stimulate blood vessel growth, such as vascular endothelial growth factor, or VEGF. And then there are also proteins that are induced that allow cells to survive oxygen deprivation, and the classic intracellular adaptation is the switch from oxidative to glycolytic metabolism that is controlled by HIF1. So now we know of over 8,000 different RNAs that in one cell type or another are induced by hypoxia and allow adaptation. And as I mentioned, the beautiful plasticity of this system is that every cell responds in a unique manner. So the HIFs send a signal, there's hypoxia, respond, and then every cell responds depending on how it's programmed. And this leads to these beautiful individual responses throughout the body. Well, it turned out that HIF1 had two siblings, HIF2 alpha and HIF3 alpha. These proteins are structurally and functionally similar to HIF1 alpha. They're regulated through the oxygen-dependent degradation, and they can also dimerize with HIF1 beta and activate transcription. And whereas HIF1 alpha is present only in is present in all metazoan species and pretty much all nucleated cells within every metazoan species, HIF2 alpha and HIF3 alpha are only present in certain cells and only in vertebrate species. So with the evolution of vertebrates, you had a much larger body mass, and this required in the specialized systems of oxygen delivery, like the respiratory and circulatory systems, to evolve. And as part of that process, HIF2 alpha and HIF3 alpha came about principally they're active during development to establish the systems of oxygen delivery. Well, I mentioned this beautiful system for regulating red blood cell production through EPO. And this system is dysregulated in individuals with chronic kidney disease. And the more severe the chronic kidney disease, the greater the deficiency in EPO production. And prior to 1986, patients required transfusions of red blood cells, and that put them at risk of various viral diseases. And so it was a really tremendous advance when the EPO gene was cloned and recombinant EPO was produced as one of the first biotech drugs in 1986. And this greatly improved the quality of life in patients with chronic kidney disease. The patients would go to the doctor and say, I feel terrible. The doctor would say, of course you feel terrible. You have chronic kidney disease. But the reason they felt terrible was that they were anemic. And when the anemia was treated, they felt much better. So this was a great improvement in the quality of life. Of course, it's an injectable biologic, which has certain limitations. And some patients, for various reasons, don't respond to EPO. So the next advance was the identification of drugs that target the protein, the HIF-1 alpha hydroxylation that I described. And these drugs inhibit the enzyme that inserts that oxygen atom, so that now there are higher levels of HIF production, leading to higher production of EPO. And this probably involves EPO production in the liver, where EPO is made in the fetus. And that probably accounts why even patients who don't have kidneys, who have their kidneys removed, respond to these drugs. So these are small molecules, so they can be taken by mouth. They increase HIF activity, they increase EPO synthesis, and they increase red blood cell production. And this is the chemical structure of the first of these drugs that underwent clinical trials, and that first clinical trial was performed here in Asia. This drug, roxadustat, has been approved in a number of countries, and there are now 5 different HIF stabilizers that have been approved in various countries for the treatment of anemia in patients with chronic kidney disease. So this was really the first translation of our basic science discoveries to the clinic. Well, the next part of the talk involves this very distinguished gentleman, whose name was Eugen von Hippel, and he had the title of Professor Extraordinary, which is quite a title, at the University of Heidelberg. And he was an ophthalmologist in the early 1900s who identified patients who had retinal lesions, and these were retinal tumors called hemangioblastomas, retinal hemangioblastomas. So he identified these tumors, and then a Swedish pathologist named Lindau in the 1920s performed autopsies on patients with these retinal hemangioblastomas and found that these patients often had tumors elsewhere in their body, particularly in the kidney. And so this collection, and this was found to be inherited through an autosomal dominant mechanism in families, and this became known as the von Hippel-Lindau syndrome. And this is a section of a kidney cancer from a patient with von Hippel-Lindau syndrome. It's called clear cell renal cell carcinoma because you can see the cells don't stain well with hematoxin and eosin. And this is because they're filled with glycogen and lipid, because their metabolism has been dysregulated, because they lack that VHL protein that I described to you that binds to the hydroxylated HIFs and causes their degradation. So in the germline, they have one wild-type allele and one mutant allele, and then in the kidney, the wild-type allele is mutated So there's no functional VHL, and that drives the tumor formation. You can see on the left the parental cells from a patient who has a kidney tumor, and you can see that regardless of whether we culture the cells at high oxygen or low oxygen, there are very high levels of HIF-1 alpha and HIF-2 alpha. But if we insert a functional copy of the VHL gene back into these cells, you can see now at high oxygen conditions, the HIF-1 alpha and HIF-2 alpha are being degraded again. So it's the failure to have the normal oxygen-dependent degradation of the HIFs that drives the formation of these kidney tumors. So we can put those parental cells back into a mouse and they will form a tumor, but the rescued cells will not. And we know now that the HIFs are activated in common cancers as well as in the hereditary von Hippel-Lindau syndrome, and that the HIFs contribute to cancer progression by turning on the expression of genes that contribute to every critical aspect of cancer progression. And so it was very exciting when a small biotech company called Peloton in Dallas developed a small molecule that can bind to HIF-2 alpha, which plays a particularly critical role in kidney cancer in von Hippel-Lindau syndrome. And when this drug, which is now called belzutifan, binds to HIF-2 alpha, it prevents HIF-2 alpha from forming a functional dimer with HIF-1 beta. So there's now no functional HIF-2 within the cells. And this was one of the first trials of belzutifan, and this is a waterfall plot looking at the change in tumor growth. The yellow bars show patients who have had a decrease in tumor growth. That's called a partial response. And then the black bars are patients whose tumors have not changed significantly, and that's referred to as stable disease. And the combination of those two, which is known as the disease control rate, was 80% in this trial in a disease where there was basically no effective therapy once the tumor had spread beyond the kidney. So this was a truly remarkable result. And within several years, the U.S. FDA had approved the drug for the treatment of renal cell carcinoma in patients with the von Hippel-Lindau syndrome. And then more recently, last year, the FDA approved belzutifan as a third-line treatment of other renal cell carcinomas in patients who don't have the hereditary form of the disease. As I mentioned, in my lab, we've been developing drugs that are both HIF-1 and HIF-2 dual inhibitors that work well in a variety of cancers. And one of the really remarkable effects of these drugs is that they take the response to the immuno-oncology drugs that you heard about from a very small number to a very high number, so that we can completely eradicate the cancers in half of the animals that we treat with the combination of these drugs. And we think that this is going to have a dramatic impact on the response to the immuno-oncology drugs that you heard about earlier. Well, I just wanted to have a few closing thoughts. The first, as we all know, is that it all starts with education, specifically science education. And unfortunately, you know that in India better than we know that in the U.S. It's sad to say. But we start with that. We do basic science because that gives us fundamental discoveries that impact on disease processes. And then if we're lucky, we can translate that into novel therapies, which if they involve small molecules can decrease costs and increase global healthcare accessibility. The focus on development of biologicals, which is very great interest now, has perhaps the drawback that these drugs are difficult to transport and store in areas without advanced healthcare systems. And so there are still many advantages to trying to develop small molecule drugs, I think. On the right is my high school biology teacher, Rose Nelson. She was my inspiration for a career in science. Thank you very much.
To stay informed about the startup ecosystem, subscribe to My Startup TV.